And so, as I mentioned before the break, we spent the first half focusing on the cancer resources that we have, and next we'll spend some time looking at the HIV and associated resources that are hosted by the team at the Los Alamos National Lab. So, with that, I would like to introduce Jenni Macke, who will present the LANL HIV sequence and CATNAP neutralization databases. Thanks very much, Jenni.
So, welcome. It's my honor to talk to you today about some of our LANL databases. We are funded by the NIH to present our databases. So first, I'm going to just take a step back and talk about databases in general, biological databases. You know, the dream that we have would be to provide a database and some scripts to go with it, where we could put in raw data, or users can put in raw data and get out just beautiful, perfect results. That's sort of the ideal, and we wish that, you know, it could all be automated and done by itself. The reality is that humans are still very much involved in the process, and I think that's been presented well by the talks about curation by the IEDB team. And our database is similar. There are steps in the process that really do require human curation. So we still try to fit the pieces together better than what any web script could do. The reality is, when working with biological database data, sometimes the pieces don't fit together as neatly as we wish they did. So again, our challenge as curators is how to fit these data into a database. So you might ask, why HIV? Why is the NIH willing to invest in a database that is only targeted at one specific organism? So, you know, HIV now has treatments that allow people who have the virus to live a normal lifespan, close to normal. There are serious side effects from the treatments, but it's not the deadly thing that it was when it first emerged. Unfortunately, not everyone has access to treatment. It's estimated that only about 77% of people who are living with HIV are accessing treatment. And there's still no cure. So the treatments have serious side effects. There's still a quest for better treatments, and certainly a quest for being able to prevent or, to prevent the disease. The other reason that we feel it's still important to have an HIV database is that it's an important model organism for many kinds of research. So obviously, it can be a model for other types of viruses, particularly variable viruses. All of what we know about HIV helped to accelerate research on SARS-CoV-2 when that emerged. It's also a very sought after organism for studies of phylogenetics and evolution, because the virus does evolve so quickly. And you can do essentially look at the evolution of the virus within a single patient. Obviously, it's an important model system in vaccinology and also in immunology. So it's a system where you can study in great detail the interactions between changes in the host's immune system in response to the pathogen, and also changes in the viral sequences in response to that immune system. So there's been a lot of in-depth study in that department. So HIV does still remain an important model organism, and that's the inspiration for our databases. So I'll be introducing the HIV sequence database and the CATNAP neutralization databases. And this is just sort of a general schematic of the layout of our databases and how they interact. So we do have a number of very, separate databases, but many of them pass data from one to the other. And the ones that I will be talking about today are the sequence database, the neutralization database, and I'll touch very briefly on a couple of our data analysis tools. So I'm one of the curators for the databases. And so the sequence database curation process starts with all the viral sequences in GenBank for HIV-1, HIV-2, CIV, and SHIV viruses. So we start from downloading all of those, and that's a fairly automated process. We also have sort of automated processes for kind of extracting the data. So all of the GenBank fields are imported. Some of those fields can go directly into our annotations, such as the country and year for sampling. Other parts of that GenBank data goes into text files, which are then reviewed for humans as to do they fit our database and which parts fit or don't fit. There are then various automated analysis processes that happen. We assign start and stop coordinates relative to the reference sequence. And that is a big part of sort of the value added that we give to these sequences relative to just a user getting them from GenBank. We mark some problematic sequences that are then easy to exclude from further analysis. Poor quality sequences have a high content of non-ACGT characters, so it's fairly easy for a script to rule those out. We also have a tool that produces kind of a rough draft of all HIV-1 sequence subtypes. Beyond that, there is a fair bit of human curation that goes into the sequences. Those subtype assignments, there's no simple tool that will give a perfect assignment. It does have to be looked at by human annotators. We have other types of problematic sequences that are a little harder to define that we look for. We do our best to link sequences with publications. So sequences often go into GenBank when there's still an unpublished paper to go with it. We do try to follow up and link it to the fully published publication once it's published. We also then look for additional data, both from the GenBank records and then from those published papers to add annotations to the sequences in our database. So in terms of what you might be able to find or what you might be able to get when you come to our database, in addition to downloading the sequences themselves, you can also get our start and stop coordinates relative to a reference sequence, subtype, isolation year, country. We almost always have those for the vast majority of sequences in the database. But there's also a lot else that's often available. We do our best to link multiple sequences from a single patient under a patient code. So that's useful in a variety of ways, both if you're looking for a whole set of sequences from a single participant, you can do that easily. And if you're looking at diversity, you can not look at those big sets from a single patient. Sample tissue, culture method, the health and drug treatment status of the patient are often annotated, but certainly not universally.
Sorry to interrupt, Jenny, but your camera has switched off again. So if you want to switch it back on.
Okay.
Much better. Awesome. Thank you.
Okay. And you can still see my slides? Okay.
Yes, we can.
So if you wanted to come to our sequence database to download some sequences, you would start from hiv.lanl.gov. This is sort of the main entry page to our databases. You can choose to go to the sequence database, the immunology database. We also have some other virus databases that are built on some of the same sort of software infrastructure. They are not as well annotated as our HIV resources, but they are there if you are working with those other types of viruses. So if you go to sequence database, This is our main menu. I just wanted to point out briefly that we do have other tutorials available under the info tab. That includes longer presentations. So our presentation today is really a fairly brief overview, but we have done longer sort of YouTube video presentations at other conferences. So if you wanted to watch other videos, they are there. If you come to our main search interface, it looks like this. And you can choose sequences based on a whole lot of different criteria. You can narrow down the sampling years that you're interested in. You can choose only certain viral subtypes. All the fields in this top box here are shown by default in the results. So you will see all of those fields in your results. You can also search for a specific region, which is very useful. So you remember we assign start and stop coordinates. So if you are interested in just GAG or just envelope, you can select that as a genomic region, or you can even just define a much narrower region using start and end coordinates within the genome. And then you will only get sequences that cover that region. There's a lot more fields here that are not shown. So there's like these little plus signs. You can open this up and choose other fields. And even if you don't want to narrow down your search with those fields, you can select them to be shown in your output. So you might put in a query and click submit. And this is just an example of a set of sequences shown on the results page. So you can see you have these are the fields here that were in that top box of fields that are always shown by default. We have the GenBank accession, the GenBank sequence name, our curated subtype, country, year, and then this genomic region map just shows you, again, where we've defined the start and end coordinates, and it shows it on a nice little map of the virus genome. You can select records in various ways. You can select all, or you can just select a range of these, or you can just select with select boxes which ones you're really interested in. The main things that I think are most commonly used from here, one is to download the sequences. The other is to make phylogenetic trees out of those sequences. So if we choose to download sequences, we get a set of choices. We can choose to have those sequences pre-aligned. So the assigned start and stop coordinates on those sequences allow us to produce a rough preliminary alignment, and you can get the sequences pre-aligned. You can choose nucleotide or amino acid output. You can clip to the selected region, which just clips the whole alignment down to just the region of interest that you put in. And you can add in a reference sequence. You can also choose to name the sequence in various ways, and that can be helpful in further analysis to have other information about the sequence in the sequence name. Another really useful little option that I'll point out is the ability to choose one sequence per patient. So if you were, for example, studying sequence diversity, you would probably not want a whole lot of sequences that all came from a single participant. So if you use this one sequence per patient selector, it will, again, it will give you one sequence per patient, and that it will be a better way to look at diversity of the virus across many people. If you were to choose to make a tree from the sequences in your search, these would be the options presented. Many of our tools, if you've never seen them before, they might look a little intimidating. You might have no idea what is Feldstein 1984. But what I will say about all our tools is if you run them with the default parameters, you will almost always get a result. And then you can go back from there and decide if you want to make different selections. The trees from our search interface are sort of rough. They're based on a fairly rough alignment, but they are very fast. So if you just want a quick look at how are these sequences related, do the sequences from one particular participant group together phylogenetically, that kind of question, you can get a quick look. If you wanted a publication quality phylogenetic tree, there are other, more lengthy procedures you would need to go through. But these are just sort of a quick way to get a look at your sequences from a phylogenetic standpoint. So I will move on now to talking a little bit about our tools. And this is our tools drop down from our menu. And as you can see, we have a lot of them. There's no way that I can give you any kind of introduction to all of them. So I'm just going to give you a brief overview and then show you two specific tools in a little bit more detail. So again, this is you can get to all the tools from the drop-down menu. If you click on this top menu where it says Tools, you can go to sort of a summary page, which gives just a very short description of each one, and that can be helpful if you're not sure, do we have any tools you might find useful? This gives you short descriptions of each of them. We've also color-coded to indicate which tools are specific for HIV or HIV and SIV in some cases. The ones in red are fairly specific to the organisms that we curate, but the ones that are in blue and purple have functionality for other organisms. Sometimes other organisms, just viruses, but often they're useful for other things as well. So we've got a whole suite of even just sort of some general bioinformatics tools. So as I said, I'm going to present just two of our tools for a quick look. This is our geography tool. Again, you would just find this on the main menu. It is a lot of fun. And so I'm not going to show a live view because it just might be too much fun and I would get distracted. So you can see a map of the world from the starting point. You can choose what range of years you're interested in looking at. You can choose particular subtypes, particular countries. And the output that you see shows you both the number of sequences in our database, and also then by the colors in the pie slice, it shows you the representation of viral subtypes in those countries. So keep in mind that this is just showing the data from our database. It's not necessarily showing you epidemiologically exactly the representation of number of infections or the subtypes of that are driving those infections. So as you would expect for the United States, it's probably the largest number of sequences, thus the largest output on the map. And then subtype B in green is the predominant subtype. On the live view, you can move around to different parts of the world. You can zoom in, zoom out, just do all kinds of fun things. So I encourage you, just if you want to get kind of an overview for the sequences in the database, I believe you can even click on the pie and go directly to a search of those sequences and get the sequences out. So this is kind of another starting point to looking at sequences, particularly if you're interested in the geography of what's in our database. The second tool that I will present is called Analyze Align. It is something, we picked this one because it might be of more interest to immunologists as well as people interested in sequences. So I'm going to show just one sort of example search that we can do using Analyze Align. And if you want to try this yourself, here on the slide, it shows you exactly what choices we have made. We are going to use a LANL database alignment, which is provided sort of already there in the tool. If you were using this for some other organism, you would paste your own sort of background alignment to analyze in the top box here. You can either paste it in or upload it. We've chosen HIV1-ONV to look at. We will take just the major subtypes as opposed to all of them to look at. We want amino acid output, and I've defined a set of positions to analyze. And those positions are relative to the residues of our reference sequence, HXB2. If you were using this for some other organism, you could specify these positions relative to the alignment columns, for example. It doesn't have to include a reference sequence. And the other thing I've selected in just for this example is in the logo options, there's a choice to mark N-linked glycosylation sites. And that's something that's often of interest in HIV research. So if, let's see, one other thing I wanted to point out briefly is that almost all of our tools have a sample input. So if you just want to run a tool and just to see, does this do anything I would ever want to do, you can just go through our tools, run the sample input for each and see what you get. So from this set of choices, this would be the output page that I would see. So we have our groups. So I asked for the output to be grouped by subtype. So these are our viral subtypes. And what we're looking at here is just subtype B. I asked for glycosylated asparaginnes to be marked. And so these O's are actually N's, but they're marked in magenta O's to show that they are a glycosylation site predicted. Below that, there's frequency by position. You can get exactly what amino acids occur in what frequency in that background alignment. And we get a consensus of that background alignment, which is shown here, and then the number of sequences that vary by what number of mutations from that consensus. So this is only like one part of the output page. We can scroll down and then look at the representations for the other subtypes. We can download these images if we want. We can see how they vary from one subtype to another. The other thing I would say is there are these gray bars in the sequence logos, and that's because the background alignment has some gaps that are not present in this subtype. So if you wanted a cleaner looking logo, if you really wanted to use this to generate logos, you could make a gap stripped alignment that would not have those gaps. So now I will move on to another tool called CATNAP. CATNAP started out just as a tool, but it's kind of taken on a whole life of its own as its own database. So what is CATNAP? CATNAP stands for Compile, Analyze, and Tally Neutralizing Antibody Panels. And the original inspiration for this was the emergence of neutralizing panels, like panels of pseudoviruses that were being used to assess HIV antibody neutralization. And so our team had the idea of, wouldn't it be cool if we could take a lot of that data and put it together and let people sort of analyze it easily. So, you know, rather than having to go get the data out of the papers and try to compare different studies, we could put it all in one place. So it is a database of antibody neutralization data, mostly IC50 and IC80 data, which is the most common, way that it's published. It is a web tool that can be used to analyze that data, and that tool can be used for other kinds of numerical data. So anytime you've got a set of variable sequences with some sort of numerical data output for those sequences, this tool could also be used to analyze those. So CATNAP brings together a lot of resources from our other databases to make this analysis more fruitful. So the curation process for CATNAP is probably more similar, will sound a little bit more similar to the curation processes that the IEDB has described. So we start from PubMed searches. Some of that is automated. Sometimes we just, in our own browsing of PubMed, we run across papers that we think might be candidates. And we look for IC50 and 80 neutralization data for anti-HIV-1 antibodies. And then once we've identified some of that data, we make choices about what to include and exclude. And I will, on the next slide, I'll show a little bit more about what we include and exclude. We get that data out of the PDF, which is sometimes a challenging exercise. Sometimes we also do obtain data directly from authors. Once in a rare while, they will offer data, but other times we make requests to them for their data. If there are new pseudovirus envelope sequences as part of that data, we get those aligned. We standardize all the names to match what's already in our database. And then we upload that data. Then there are some tools that make that data more useful. We provide pre-calculated geometric means. So if there are multiple studies that looked at a particular antibody with a particular virus, we will provide a mean of that data, and then it's up to the user if they want to use data from one particular study or if they want to use those means. We provide pre-calculated genetic signatures. And then the CATNAP database also pulls in a lot of data from the HIV sequence database, such as the sequence subtype, country, year, etc. It pulls in data from our antibody database about the binding region and other things about the antibody. It pulls in antibody contact sites from our feature database. So there's just a whole lot of data sort of brought together to be more useful. So what are we looking for in the data for CATNAP? So we're looking for pseudovirus assays only. We don't include live virus assays. The data we're looking for typically looks like what's shown in this picture, where you've got 7 antibodies here assayed against a panel of pseudoviruses. And this is showing just IC50 data. So it is the type of data we would be looking for. We're looking for novel data. So studies will often include previously published data as a comparison to their new data. So we try pretty hard not to re-upload data that came from a previous study. We would exclude assay values that seem very, very different. So sometimes they will be assaying a virus that has an antibody and a set of viruses that was previously studied, and their values are just completely different. We would rule that out. We don't take non-standard assay types or cells that were, assay cells that were grown under selective condition. One good thing about the HIV field is that there are published standardized assessments and standardized assay protocols that are produced by David Montefiore's group. And we work together with them to keep those protocols on our website. We can't always know when we look at a particular study if they used exactly that set of protocols, but we will look for consistency with those protocols in their research methods. So if you wanted to explore CATNAP, you would find it on our main menu under Tools, and it's also under Databases because it is both. And when you come to it, you would see several choices. There's the main CATNAP tool, which is the full database with the analysis capability. We do have full database downloads available if you wanted to just get the neutralization data and work with it on your own. And so that's probably the most commonly used part of this. There's also this custom input that I will mention. So this is the area where you could put in other kinds of data. So any set of variable sequences together with some kind of numerical data could be analyzed using this tool. And then the hybrid version uses the database data to compare to your own new data, say. So the main CATNAP tool input page, this is it. You can choose what to analyze in a whole lot of different ways. You can choose, if you know the name of a particular study you want, you can go and find that study. If we don't have it, you're welcome to ask us to add it, or ask us why we excluded it. You can select specific antibodies by name. You can also select the antibodies, say, by their binding region, or you can just type in a list, and that's often the easiest way to do it, is just put your own list in. You can choose, again, the pseudoviruses that they were assayed against by various criteria. You can just select all of them, or you can select them by subtype, by well-known panels, etc. And then you can choose what you want to see. So maybe you just want to see the assay data. You choose assay, and it will just show you the assay data that we have in our database. You can choose to see all of the details about the antibodies, all the details about the pseudoviruses, or you can choose to look at the analysis page. Now, I'm not going to show the analysis page right now because it is going to be part of Jennifer Mamrosh's talk after mine. So that's coming up. I just wanted to highlight some of the additional fields of data you can get out of this neutralization database, in addition to just the IC50s and IC80s. We store heavy and light chain sequences for as many of the antibodies as we can. We store data about the germline genes for the human antibodies and their degree of somatic hypermutation, and we report those as reported by the authors in the published papers. Some of the basic data from our antibody database, like the binding region, the isolation paper, etc. And then again, the contacts and other envelope-associated features are pulled in from our feature database. You can also get lots of details about the pseudoviruses, their sequence, of course, their GenBank accession, infection stage information, and a link to the participant information and other sequences from that participant. So please enter any questions in the Q&A, and if you have questions about what that CATNAP analysis is going to look like, hang on, because Jennifer Mamrosh will be showing that. And please feel free to use the email addresses on our website to contact us. We are a service for our users.